A study of the cytoskeleton of malignant cancer cells involving F-actin, GFAP, vimentin, and microtubules. Cancer is a disease renowned for its aggressive nature and the problems in treating it. Two major problems with tackling cancer are its ability to metastasize and the fact that cells causing the disease are our own body cells, and so it's hard to create a drug that will differentiate between healthy cells and cancerous cells. There are over 200 different cancers, so another problem is finding a cure that could work on them all. There are, however, 6 hallmarks of cancer common to all, and one of these is metastasis, and so targeting this could result in effective treatment against all cancer. Investigated the cytoskeleton because it's required for cell motility and is therefore vital in the process of metastasis, which is one of the 6 hallmarks of cancer. Because of its requirement in this process, it may be that cancerous cells have an upregulation of the cytoskeletal components, and therefore the cytoskeleton could be targeted to differentiate between healthy cells and cancerous cells. Alternatively, the cytoskeleton could be targeted to prevent metastasis from occurring by preventing cell motility. There are 5 steps in metastasis once the tumour is developed. Migration occurs, then intravasation, then transport, then extravasation, and then metastatic colonization. As the cytoskeleton is involved in cell motility, it's required for all of these stages. The cytoskeleton is comprised of 3 fibers: microtubules, microfilaments, and intermediate filaments. Microfilaments regulate the cell shape and movement of the cell surface. Intermediate filaments bear tension, providing support, and microtubules help to maintain the cell shape and provide a route for the organelles to move throughout the cell. I have identified the cellular localizations and quantitative expression of microtubules, F-actin, which are microfilaments, GFAP and vimentin, both intermediate filaments, using flow cytometry and immunocytochemistry. From the data collected, it is clear that the function of the protein is directly related to the level of expression and their localizations. GFAP and vimentin are both present throughout the cells to aid in cell structure. Microtubules are also present throughout the cell and provide a route for organelles to move along. F-actin is concentrated in the periphery of the cells as it regulates the movement of the cell surface. These localizations also support ideas of how the cytoskeleton aids in intravasation. Microfilaments being present at the periphery means that they can create protrusions which push into the holes in the basement membrane of a blood vessel. Microtubules are present throughout to enable enzymes to move the tip to continue movement through the membrane, and intermediate filaments are also present throughout to provide strength and support to the protrusions. Due to this way of action, I think that microfilaments would be best to target, as this will prevent protrusions from forming and therefore prevent the cells from entering the bloodstream and being transported around the body.